coming to today's Immune Zoom seminar. It is my great pleasure to introduce our speaker, Dr. Anna Molofsky, who is currently an associate professor at UCSF and the Samuel Barandas Endowed Professor in Neurobiology and Psychiatry. Anna is a pioneering neuroimmunologist whose work bridges molecular neuroscience, immunology, and psychiatry to uncover how immune signals shape brain development, plasticity, and disease. With a distinguished academic background, including a bachelor from Amherst College, an MD PhD from the University of Michigan, and postdoc training at UCSF. Anna has been made groundbreaking discoveries about the role of microglia and cytokines in synaptic remodeling, memory formation, and behavior. Her lab's innovative approach, which she described as studying neuroimmune circuits one cytokine at a time, has revealed how immune cells like microglia and the innate lymphocytes directly influence neural circuits, with implications for neurodevelopmental disorders, neurodegeneration, and mental health. Beyond her research, Anna is a dedicated mentor, actively supporting trainees at all levels. Her work has earned her numerous prestigious awards, including the Pew Scholar Award, the NIH Director's New Innovator Award, and the Daniel Efron Research Award. There's a long list, and I wouldn't read through. So today, I think Anna is going to share with us about her lab's recent discovery on the cytokine regulation on brain function. Without further ado, Anna, the floor is yours.
Thank you so much, Chalet, for the very kind introduction. Happy to be here. Go ahead and share my screen. So, as you heard so beautifully introduced, what my lab works on, how cytokines regulate the healthy and the diseased brain. I will add that in addition to what was mentioned, I'm also an adult psychiatrist, so I don't see very many patients anymore. My clinic, my practice is small relative to my research lab, but a lot of what drives my interest in how the immune system affects the brain is wanting to understand, unexpected regulators of brain function that many neuroscientists may not be thinking about and that we should be thinking about. I think all of us here at an Immune Zoom appreciate the power of immunotherapies and can recognize that we need to be thinking about immunotherapies for neurological disorders very seriously. So, for example, the finding that Epstein-Barr virus increases the risk for multiple sclerosis at the same time that...
Sorry, Anna. Are
Sir.
you sharing your slides right now?
Yeah, I am. You can't see them?
I can only see actually the...
Oh, I see. Okay, here, let me unplug external monitors to make sure there's no problems. Thank you for catching that.
Sure.
It is sharing the wrong thing, that's why. Let me share again. I'm going to share my desktop. Okay. There we go. Immune Zoom.
Okay, excellent.
Take two. I'm going to skip over the slide. We already did it. All right, so let me tell you why we should care about the immune system in neuropsychiatric disease. So we know from keystone neuroimmune diseases like multiple sclerosis, that it's worthwhile to understand how the immune system affects the pathogenesis of disease because it can lead to new immunotherapies. And multiple sclerosis is a great example of that because B-cell immunotherapies and other immunotherapies have really revolutionized the treatment of this disease. There are many other diseases for which viral infections. Now, in the case of multiple sclerosis, Epstein-Barr virus, which is this larger virus, increases the risk for MS 30-fold. For many other post-viral neurological syndromes like West Nile or what we think of as long COVID, Here you can see SARS-CoV-2. We still don't understand what's the relationship between immune activation and the changes in brain function. Alzheimer's disease and neurodegenerative diseases in general are another huge area where people are beginning to appreciate how important the immune system is in the context of brain disorders. And here you can see, for example, 6 microglia, which are brain-resident macrophages surrounding an amyloid plaque, which is a canonical pathological feature of Alzheimer's. Schizophrenia, a disease that I would love to, if we could understand more about. The top hit in the genome-wide association studies for schizophrenia is the MHC locus. It says MHC1, but class 1, class 2. And that suggests that something about the way that we interact with our immune environment is critical to some subgroup or some aspect of schizophrenia, although I would say we don't know how. It's not like some diseases where we know which HLA specifically increases the risk, or even if there is a specific one. So there's a lot still to understand. But one general model of how neuropsychiatric diseases in general may depend on the immune system has to do with the idea that the brain has very sensitive periods of development. And I'll talk a little bit about that here. Many have suggested the concept of maternal immune activation. So, for example, that maternal sickness could lead to cytokine production that crosses the placental barrier and affects the developing brain, which expresses many cytokine receptors. This is a concept that needs to be tested specifically, but it comes from a lot of literature suggesting that there are links between early life infections and the later development of psychiatric diseases, including schizophrenia and many others. So what my lab has sought to understand is, what are these receptors that are expressed in the brain, and how do cytokines mediate the function of the healthy developing brain? And so just to put it in context, what do we mean when we're talking about the developing brain? In humans, the brain continues to develop until you're about 25. It begins with the birth of neurons, the growth of these very long, very unusual cell types that have thousands and thousands of synapses. And after neurons have positioned themselves in the cortex and grown, they begin to form synapses, and this can take years and years and years. So these green spots here are excitatory synapses. They are the synapses that encode information. And these little blue spots that you can see, this is the same neuron, has over 8,000 excitatory synaptic connections and over 1,000 inhibitory synapses. These are constantly being remodeled as we learn from experience. And cells like microglia, brain-resident macrophages, and astrocytes, which are the kind of the fibroblasts of the brain, play a critical role in this remodeling process. So you can see that just like any other tissue, like the lung or the liver, there is regeneration and remodeling happening in the brain, but we need to think about it in terms of neurons and synapses. So how do we begin to think about the role of the immune system in the brain? Well, we know about these 3 major arms of the immune response, type 1, type 2, and type 3, which were defined in the setting of the pathogens that they tend to elicit, right? So the immune system is known primarily as a pathogen defense system, especially for most neuroscientists who may not be thinking about neuroimmunology as much. Antiviral, allergic, and type 3 responses. And each of these responses has many different cytokines. I'm oversimplifying here, but interferons, type 1 and type 2 interferon, drive type 1 immune responses. IL-4/13 are some of the key drivers of allergic inflammation, and IL-17 in terms of the type 3/17 response. And we know that in some settings, too much cytokine activation is pathogenic, such as in the case of psoriasis, where IL-17 blockade has been, essentially curative for that disorder. Similarly, in asthma and eczema, there's trials to try to block IL-4/13 signaling. But in many cases, most cases, cytokines also have physiologic roles, right? So they are maintaining a homeostatic state, right? Healthy immunity is not an absence of immune response, it is a balance of immune responses. So this figure that I use here is adapted from a review by Maya Kodas and Rich Loxley, where these little squiggles here are actually springs, indicating that there's this constant tension between this acute, alert, and restorative state of the immune system that maintains this tissue homeostatic state. And this is what we want to understand when we're thinking about how the immune system regulates the healthy brain. For example, my colleagues Gloria Choi and Jun He have done a lot of beautiful work showing how IL-17 impacts the brain. And today I was going to talk about two studies from my group, one of which has focused on the role of type 2 immune responses, and specifically interleukin-13, in promoting a type of synapse development. And in the second part of the talk, I'm going to talk about a completely different function of how type 1 interferons are promoting the elimination of certain neurons in the developing brain. And in both of these cases, these are not pathogenic cytokine responses. These are adaptive and homeostatic cytokine responses. And our goal, if we were to think about these in terms of immunotherapies, would be to harness these responses in order to help build the brain. So the first part of the project is a collaboration with Ari Molofsky, led by Jerrica Barron and Nick Mraz, two very talented graduate students. And it began with an interest in understanding not microglia. At the time, everybody was working on microglia, but understanding the immune cells that sit just outside of the brain in this region called the meninges. So there are no lymphocytes typically within the brain, but there are lymphocytes in these meningeal regions just underneath the skull that can secrete cytokines that circulate throughout the cerebrospinal fluid, which you can see here in blue. So the brain can be bathed in cytokines. We don't fully understand yet how the brain barriers work and which proteins can come across and which can't. But we know that the cytokine receptors are inside the brain and the cells making the cytokine are outside the brain. So I'll tell you a little bit more about that. We began because we're interested in development. And we know that there's lymphocytes in the adult meninges. We wanted to examine what the lymphocyte populations were in the developing meninges. So here you can see non-B lymphocytes by single-cell sequencing from postnatal day 14. And one thing you'll note is that adaptive immune cells like T cells are relatively minor contribution at this time. But innate immune cells are very abundant, including group 2 innate lymphocytes. So these are major players in allergic immune responses. They're kind of the innate counterpart of Th2 cells, but their role in the developing brain was not well known at that time. One of the major cytokines released by ILC2s is interleukin-13. And so we looked to see whether these meningeal ILC2s were making IL-13. By flow cytometry, you can see a reporter here. This is the SMART13 reporter, so-called. You can see that they produce quite a bit of IL-13, but only during development. So there's this burst of IL-13 production. Here you can see CD4 cells, for example, are not producing any at this time, which shouldn't be a surprise. So there's a burst of IL-13 production during early life, raising the question of whether this time period, which is a very important period for synapse development, might be sensitive to ILC2s or IL-13 signaling. You can see here, as I alluded to before, that these cells are present in the meninges. So here we've taken this surface. It's just like the pericardium or the peritoneal, the peritoneum. It is the membrane that surrounds the brain. If you put it flat on a slide and look at it and look at IL-13 expressing cells, you can see that they are increasing in the meninges over the course of early development. And there are no IL-13 producing cells inside the brain at all. In fact, this is the same amount of space as you see here. So you can see how abundant IL-13 producers are in the meninges, but not in the brain itself. So anything that I show you about the effects of IL-13 definitely suggests that these are meningeal signals and not brain-derived signals. So we used, in this study, a genetic model to deplete ILC2s, whereby we bred IL-5 Cre to Rosa DTA, which leads to about an 80% depletion of ILC2s. We now have, there are better tools, such as one, I don't know, better, but different tools built, such as one built by David Ardis's lab, and Amur-Cree, that can delete ILC2s as well. This is the tool we were using at the time. And then we asked, well, what happens when you ablate ILC2s in the developing brain to synapses? So this is how we quantify synapses. Synapses are less than half a micron in size. So, what you're seeing here is synapses that are identified by the co-localization of one neuron touching the other. So we can't see the cells, we only see where the point of contact is. And when we see both a presynaptic and a postsynaptic marker, VGAT and Gefrin, that's what we know is a bona fide synapse. So this is the strategy we've used throughout much of this paper. What we found is that when you deplete ILC2s, you have a loss of inhibitory synapses. This is a very different phenotype than what we saw with interleukin-33. It's another set of studies I won't talk about today. But this was very specific to inhibitory but not excitatory synapses. And when we deliver ILC2s directly into the ventricular system of the brain so that their IL-13 presumably can circulate throughout the CSF, we see an increase in inhibitory synapses. And these changes may look small compared to what one might expect if you're infecting with a pathogen or injecting LPS, but in the setting of brain synapses, these are really robust effects. And as I said, excitatory synapses are not altered. So what about IL-13, which I've been alluding to? There's this burst of IL-13 production by ILC2s. We looked in IL-4 receptor knockout animals, which can't sense IL-13, and we saw the same decrease in inhibitory synapses. And perhaps what's most exciting to me, Jerica injected recombinant interleukin-13 directly into the ventricle of the brain, and less than 24 hours later, examined inhibitory synapses and saw that it was sufficient to increase inhibitory synapse formation. This is the kind of experiment that makes me wonder about potential therapeutics, right? Cases in which you might want to increase inhibition. And finally, we wanted to understand what cell type is sensing IL-13 from ILC2s. Is it microglia, as we had shown in previous work with IL-33, or could it be neurons or some other cell type? And the answer turned out to be neurons. So when we delete the IL-413 receptor using VGAT-CRE, which is a marker of inhibitory neurons, these are the neurons that form inhibitory synapses onto other neurons, we completely phenocopied the effects of deleting ILC2s or deleting IL-13 globally. So that suggests that IL-13 is signaling directly to these neurons to help them to form more synapses. And in terms of behavior, I just want to point out the one that had the most robust effect in our study, which is a assay that's been used to study social behavior. This is called the three-chamber social interaction test. A mouse is placed into a chamber that has a mouse under one cup and an empty cup in the other chamber, and the mouse is allowed to freely explore. And you see that mice tend to spend most of their time around the other mouse because they're curious about the mouse and they're social animals, and less time in the space where the empty cup is. Now, when we ablate ILC2s, we see that this preference goes away. So you can see a decreased preference for the social cup. And the same happens when we delete the IL-4 receptor from inhibitory neurons. So I'll summarize this part of the talk by saying that there's a type 2 immune circuit mediated by ILC2s in the meninges of the brain that signal directly to inhibitory neurons that express the IL-413 receptor to promote their ability to form synapses onto other neurons, that this effect is isolated and enriched at this particular early life window of development of synaptogenesis, because that's when IL-13 is produced. That increasing ILC2s or IL-13 can cause more synapses to form, that deleting ILC2s or the IL-13 receptor can decrease synapses, and that these effects that are happening in early life can have a long-lasting impact on social behavior, even into adulthood. It raises interesting questions, such as whether, if we think about how much of the developing world has a high parasite burden, where type 2 immune tone is much higher, whether that might lead to more inhibitory synapse formation, whether parasitic infections could perhaps affect cognitive development in children. We don't have evidence of this, but these are the things I worry about. It also raises the question of, regardless of cause, could IL-13 be a potential therapeutic strategy to promote inhibition in situations where you might need that, for example, epilepsies? And I just want to point out, and this was what I was trying to pull in just as we were getting the talk ready. A paper just came out that found actually a very similar thing. So that's always really exciting to see when you find something. And another group has deleted group 2 innate lymphoid cells using an MUR-CRE strategy, I believe, and saw effects on inhibitory synapse formation as well as on behavior. So that's been exciting to see. So in the second part of my talk, I want to switch gears a little bit and talk about type 1 immune responses, and our finding that type 1 interferons in particular promote the elimination of certain neurons in the developing brain. And for this part of the talk, I'm going to introduce microglia, which I've alluded to before. These are the only immune cells within the brain parenchyma. Like I said, there are innate lymphocytes and all kinds of lymphocytes in the meninges, but within the brain itself, 10% of the cells in the brain are microglia. And what I showed you in this movie, I'll play it one more time, is that the brain is full of synapses, and here you can see them in red. These are excitatory synapses. And much of the synaptic protein, not much, but can be found inside of microglia. So microglia remodel the brain, they take up synaptic proteins. And so they are critical to how neural circuits form and remodel. Leah Dorman, who is a graduate student in my lab, wanted to understand what the triggers were that drive this microglial remodeling. And so she designed an experiment, and a lot of this work was done in collaboration with Carolina Skubas, a postdoc in the lab. Leah designed an experiment to trigger remodeling in the brain. And what you're seeing here is the whiskers of the mouse. So the blue whiskers, the right side, connect to the left side of the brain. The red whiskers connect to the right side of the brain. And when you lesion the whiskers early in life, postnatal day 2, each one of these so-called barrels represents a single whisker. There's a topographic map of the whiskers in the mouse because the whiskers are so important. Basically, like a third of its cortex is devoted just to sensing whiskers. If you lesion whiskers in early life, the whole shape of this region of the brain changes to match the new topographic map. And when we did this, Leah did the experiment and then collected microglia a few days later to understand how do microglia respond to a remodeling of the brain. And here's what she found, and here's a single-cell-seq plot. Only one population, there were small changes in many populations, but the one that was most strikingly altered is this cluster 8, which was defined by the expression of type 1 interferon response genes, including this one called IFIDM3. So there's an interferon responsive microglial state that expands 20-fold in this remodeling brain setting. And here's what they look like in the brain. You can see here that these cells, which are labeled in red with the marker IFIDM3, are very rare. But when you deprive whiskers, they expand. They're still a rare population, but they expand after deprivation. And what's most striking is when you look at individual cells, so unlike that lacy microglia that I showed in the video earlier, these cells are highly phagocytic. So this cell has a very distorted nucleus, and it has two processes. One of them is eating another cell, which is a neuron, which I'm not showing you the data here. And the other process is in the midst of digesting a neuron. So these cells are hyperphagocytic. Not only are they all phagocytosing, but when they are phagocytosing, they are typically eating 2 or 3 cells at a time. So interferon responsive microglia, specifically type 1 interferon responsive microglia, are engulfing entire neurons in the developing cortex. This is not remodeling synapses. This is whole-scale elimination of other cells. And here's another example of that kind of phenotype, looking in zebrafish, which is a completely transparent until about 14 days of life and can be a really useful model for live imaging of the developing brain. In red, you have neurons. In green, you have microglia. And you can see how microglia make these surveillance contacts with neurons. So it's contacting the neuron, and then it's going away again. It's like microglia are constantly checking out if the neuron is okay, right? Now, in the setting of poly-IC, when we drive, type 1 interferon responses, you can see that the microglia become highly phagocytic. This neuron in the dotted line here becomes engulfed. I'm just going to put my pointer on so I can point a little better. And so you can see that in the fish as well, you have a type 1 interferon responsive subset that is capable of eliminating neurons. So to study this and figure out the cell types involved and the mechanisms behind it, we return to the mouse model. I'll just remind you that type 1 interferons, including interferon beta and multiple different alphas, all signal through this receptor, which requires IFNAR1. That's an obligate part of the heterodimer. And by deleting IFNAR1, we can see whether it matters to the developing brain to have interferons. And here you're looking at a single microglia from a IFNAR1 deficient mouse. The yellow dotted line indicates the nucleus of the microglia, and each of these arrowheads indicates a phagosome. These are large, distended phagocytic compartments that have DAPI-positive material inside them. So what we think is that microglia from type 1 interferon receptor deficient mice, in other words, microglia that are unable to mount an interferon response, have defective phagocytosis. They're able to phagocytose, but then the cells get stuck inside of them. We called these bubble microglia simply because of their bubbly appearance, in honor also of Francesca Perry, who had first observed this kind of a phenotype in the fish. And we quantified bubble microglia as being those microglia whose phagosome is larger than its own nucleus. And we find that indeed in type 1 interferon deficient mice, there is this peak of bubble microglia at around day 5, which coincidentally is the age that we did our single cell sequencing. But by adulthood, type 1 interferon deficient microglia look normal again. So there's something very unique about this developmental window where it's really important to be able to mount an interferon response. And you can see here then when we delete the interferon receptor specifically from the myeloid lineage, we recapitulate this bubble microglia phenotype. So microglia are sensing type 1 interferons. What happens to the neurons? So many people asked us this. They asked, well, maybe there's dead neurons and the microglia are helping to eliminate dead neurons. And I will tell you briefly that that is not really clearly the case. What we did find is an accumulation of neurons that were DNA damaged, which we're interpreting as stressed neurons. So here you can see a marker called 53BP1, which is usually diffuse in the nucleus of cells, but when there's a double-strand DNA break that needs to be repaired, 53BP1 forms these bright spots here. So here you can see 3 to 4 neurons, each of which have a double-strand DNA break in them. And what we observed is a really robust increase in the number of double-strand DNA damaged neurons when in the absence of type 1 interferon signaling. So these microglia, we hypothesize, are helping to eliminate neurons that are damaged or under stress. And I want to remind you in the setting of type 1 interferon signaling that most cytokines are working in concert with pathogen or with molecular patterns that help to define the functional state of a cell. So, for example, in the setting of microglia, I showed you that type 1 interferons are a signal that can help instruct a particular microglial trait. But microglia are also constantly sensing the brain environment. They can sense neurotransmitters like GABA, they can sense ATP, and they can sense molecular patterns associated with interferon responses, which typically are nucleic acids like DNA or RNA. And that's what I'll talk about in the next couple of slides. But it's the combination of those two types of sensing via pattern recognition receptors and cytokines that lead to these different functional states of microglia. We have microglia that are engaged in the remodeling of synapses versus microglia that are capable of engulfing entire neurons. And we hypothesize that type 1 interferons, in combination with molecular patterns in the tissue, help to drive microglia towards this neuron engulfing state. So what are those molecular patterns? This tends to be in the setting of interferon responses. These are very well defined. DNA nucleic acids, in short. So we can have Caroline in the lab got multiple different knockout mice to understand which molecular patterns might be driving these type 1 interferon responsive microglia. One of these is MAVS. So this is involved in cytoplasmic double-stranded RNA sensing via the RIG-I MDA5 pathway. So in other words, when cytoplasmic double-stranded RNA is present, this pathway can drive activation of the interferon response, including production of interferon alpha beta. Double-stranded DNA is, in contrast, is sensed through the cGAS STING pathway. And extracellular double-stranded RNA in the ambient environment is sensed via this endosomal pathway and detected by TLR3. There's more, obviously, but these happen to be the 3 pathways that we knocked out. And then Caroline looked for the presence of bubble microglia, the same type that we saw in interferon deficient mice. And interestingly, she found that MAVS deletion, in other words, sensing of cytoplasmic double-stranded RNA, is critical for brain health. In the absence of double-stranded RNA sensing, we see an increase in bubble microglia. We see an increase in neurons with double-strand DNA breaks. But knocking out cGAS or TLR3 had no effect on these phenotypes, suggesting that double-stranded RNA is the physiologic sensor in this case, thing being sensed. What's interesting about that is that right around the time we were preparing revisions for this paper, this very lovely study came out from Ha-Chung Chung's lab, showing that neurons are one of the major producers of double-stranded RNA, like all over the body. Neurons have abundant amounts of double-stranded RNA, and that's thought to be because they have very long 3' untranslated regions. So their mRNAs generate a lot of secondary structure. And so we thought that it makes actually quite a lot of sense that if microglia are sensing damaged neurons, that they might be sensing double-stranded RNA. And when we perform this whisker lesion, we're causing neuronal stress that leads to the release of double-stranded RNA and drives type 1 interferon responsive microglia. And just to give you a quick sense of how this affects behavior and development of the cortex, I will briefly summarize that by saying that the layer of the brain where we found these microglia is very important for basically helping that region of the brain communicate with other parts of the brain. What are the whiskers sensing on the other side? How do we connect all that information together? Here it is. It's called layer 5. And what Caroline found is that when we don't have interferon responsive microglia, we have an accumulation of neurons in layer 5. So indeed, it seems like these microglia are necessary to eliminate neurons during development, right? We don't need as many neurons as we're born with. We need the right combination and pattern of neurons. And this actually, interestingly, has a functional impact as well. So in this assay, this is a very simple behavioral assay that you can do with mice. And what Caroline is doing is she's tickling the whiskers of the mice to see if it annoys them. And this mouse is not bothered. It's eating a little poo, it's smelling, it's interested in the stick, right? It doesn't have an aversive response. Now, she noticed that other mice would get very upset. They would try to bite the stick or run away from the stick or even try to climb out of the cage. And that's called tactile hypersensitivity. And interestingly, when the data was unblinded, it turns out that type 1 interferon deficient animals have a big increase in tactile hypersensitivity. And this is phenocopied when we knock out IFNAR1 only in the myeloid lineage. And interestingly, this imbalance between excitatory and inhibitory neurons, which I showed you before, and this increase in tactile sensitivity are two phenotypes that have frequently been observed in neurodevelopmental disorders like autism. So it's worthwhile to really understand why the immune system, why an immune molecule like type 1 interferon might be relevant here. I'll summarize this part of the talk by saying that type 1 interferon responsive microglia in the developing brain serve a critical function. One thing that's very surprising about this, of course, is that we usually think of type 1 interferon responses as antiviral responses, right? There is no virus involved here. In fact, all of our studies compare ipsilateral and contralateral hemispheres of the same brain, right? So these are physiologic type 1 interferon responses. Type 1 interferons are produced in very, very small amounts, tonic interferon signaling in the brain. We don't know what cell type is making them, but we know that there's just enough to make a microglia competent to respond to these stress signals, such as double-strand RNA from neurons, and drive the emergence of a cell type that's capable of eliminating some neurons from the system, and that this is really important for maintaining excitatory inhibitory neuronal balance and sensory behavior. And like I said, this makes sense if we understand that neurons have a lot of double-stranded RNA intrinsically. And it also fits nicely with, in another way, with a study that came out suggesting that TLR9, which is a DNA sensing pathway, is important for learning and memory in the brain. So there's an emerging theme that nucleic acids, which presumably drive interferon responses, or in some cases may act independent of interferon responses, play a critical role as maybe stress signals in the brain, or what we are calling brain-associated molecular patterns. And I just want to point out as well that interferon responsive microglia are present across brain pathologies. So in this plot, Leah took our own type 1 interferon responsive data set, I think about 80 genes, which was our core interferon responsive signature, and compared it a bunch across many different single cell seq data sets that are out there. And what you can see here is that there's an enrichment of type 1 interferon responsive microglia in Alzheimer's disease mouse models, really robustly in glioblastoma, obviously in mouse models of viral infection like SARS-CoV-2, but also in aging and in stroke, in settings where you might not necessarily expect sterile damages, where you wouldn't necessarily expect to see interferon responses. So it's a highly conserved response. Here you can see in a collaboration with John Weinstein's lab at the University of Washington, you can basically tell where the stroke happened in the mouse brain based on its IFIDM3 staining, very robust interferon responses in the setting of stroke. And this raises a broader question, which is that if, even the healthy brain has interferon responsive cells and needs interferon responses for brain development, what happens if you have too much of a good thing, right? So in the setting of an actual viral infection, you could imagine that interferon responses would go through the roof in the brain, that you would get interferon responsive microglia, and perhaps they're even able to eliminate neurons. And so, it occurs to me, thinking about this, about post-viral neurological syndromes like long COVID, I had a lot of skepticism about the idea that long COVID could affect the brain, at least early in the pandemic. And one of the things that changed my thinking on this issue is this study from the UK Biobank. And the UK Biobank is a treasure trove of data because there is structural brain imaging data along with, blood proteomics and all kinds of data from a large cohort of people. And in this case, they were able to observe, they had imaged people before and after they had COVID in the first wave, before the vaccine was available, and observed changes in brain structure. In other words, there really was a decrease in brain size, predominantly in the areas connected to smell. As you recall, in those first waves, there was a lot of damage to olfactory neurons, but in other brain regions as well. So this makes me wonder whether indeed interferon responses could lead to the loss of some cell types in the brain and whether this is worth investigating further. So here's an example from our own group using a particular model of SARS-CoV-2 infection, which perhaps is not the best model because it's expressing a recombinant human ACE2 receptor. In this case, we see that there's a lot of SARS-CoV-2 infection of neurons in collaboration with Raul Andino's lab, and we also see microglia surrounding and potentially engulfing those virally infected neurons. And I want to return to the theme of brain development and how important it is to understand the immune system in brain development and show just a little bit of unpublished work that my group has been thinking about to understand again how viral infections might affect the developing brain. So early life viral infections, as I alluded to earlier in the talk, can have very long-term impacts. It's thought that in early life, not only is the immune system developing, and so it's reliant on innate immunity, adaptive immunity hasn't come online, but the brain is also developing. So these two systems are developing at the same time. And so it's thought that viral infections during this early window of life can be particularly impactful. And may increase the risk for neuropsychiatric diseases. What we're wondering is whether these early life infections, by leading to the emergence of interferon-responsive microglia, might cause a hit-and-run damage to the developing brain. In other words, the immune response returns to normal, but neurons have been eliminated, leading to long-lasting sequelae. And so Richard Jin, who's a postdoctoral fellow and a psychiatrist in my lab, has developed a model of early life viral infection in collaboration with Raul Andino's lab, where it is observed that the virus actually does proliferate within the brain. And here you can see mice are infected intragastrically in early life, but we get quite a bit of viral proliferation in these hotspots of the brain. You can see in purple here, these are IFID-M3 responses indicating interferon responsive cells and viral pockets. And if you zoom in on those, we see that over the course of many, many experiments, these patches of interferon responses happen in specific regions of the brain, including the cortex. And by the peak of infection, when the mice get sick, we can see robust interferon responses in the brain. And within those patches, we can see that they're concentrated in certain brain regions. Now, what's exciting about this model is that the mice go on to fully recover, and by 30 days later, interferon responses are back to baseline. And yet, during the peak of infection, we can observe that there are interferon-responsive microglia within these hotspots in the brain, and they're highly phagocytic. So here you can see an interferon-responsive microglia, which I've outlined with the red dotted line. You can see that it's eaten 2 cells. We don't know if these are neurons or what exactly they're eating. This is something that we're trying to figure out, but it suggests that interferon responses and highly phagocytic microglia are present in this viral infection model. And what's more interesting is that if we follow these mice over time, 100% of them recover. There's no mortality. After they get sick, they grow up and look just like their uninfected counterparts, but there's long-lasting changes in behavior. So here's an example of a behavioral assay that we do to test sensory stimuli. I showed you one where we tickle the mice and we look for an aversive response. This is kind of the opposite. In this case, the mice have a choice. They can either go through this fuzzy cloth tube. It has like a little blanket that Richard bought on Amazon that feels very soft and nice, or they can go through this naked tube. And here's what it looks like. The mice are just freely observed. What Richard found is that mice that have been previously infected have a preference for the soft tube. This is called gentle touch preference. So there's long-lasting changes in sensory behavior in these animals that have been previously infected with the virus. And it raises kind of an interesting question, right? How do our immune experiences shape our adult selves? How does viral infection in early life, all of the kind of immune exposures that we have as we're growing up and developing, impact our brain, right? We think a lot about how our mothers and our fathers impact our brain, how our schools impact our brain. But in fact, there may be many aspects of our environment that we don't think about that are also impacting the way that our brain develops. This is not necessarily a bad thing. It could simply be a feature of your personality, whether you like a fuzzy blanket or not. But it's interesting to note that the brain and the immune system are maturing together. And just one more note that I would make before wrapping up my talk, and I think this is my last slide. What I showed you is that type 1 interferon responsive microglia engulf whole neurons. And I showed you also that microglia have a tremendous capacity to remodel the brain. And one thing that I think is important to note for anyone in the audience who's heard this idea of microglia synaptic pruning, which is basically the idea that microglia might engulf synapses like this. I want to sort of broaden your perspective. If you have time and you're interested, please check out this review from September of 2023. It's very short, and in there, I really talk and a little more in depth. I wrote this together with my colleague Ukpong Ayo about what are microglia actually doing in the brain. The idea that they're eating synapses is an interesting one. It may not really be supported by live imaging data. They may do this in some scenarios, but in many cases, we think that microglia have much more complex functions, such as when they can eat entire cells. Obviously, if a whole neuron is eaten, its synapses will also be eliminated. In other studies, which I didn't show you today, we've shown that microglia have a really important role in remodeling the extracellular matrix. When you remodel the matrix outside of a cell, this can cause synapses to disappear or be shed, and is another mechanism by which microglia affect synapse numbers. In many cases, microglia can even promote the formation of synapses. So it's really important for us to be sort of mechanistic and curious about the many different ways in which microglia can remodel the brain, and to identify specific cytokines that may have distinct roles in these different pathways. So I'll stop there for now. Thank you so much to whoever is watching out there. I don't know who's there. Thank you to my lab and all my collaborators, my funding. Happy to take questions.